Good afternoon, and thank you very much for organizing this webinar. And the idea of this webinar is really to increase the awareness that sometimes livers which are considered as waste can still be used to save life and to develop advanced therapy medicinal product, which is now a reality, a clinical development for treatment, in this case of acute and chronic liver failure, but of a number of diseases. So this is still a clinical development, but clinical development means that patients are being treated within clinical trials to demonstrate that this therapy can also save life. And the idea is really to avoid competing with organ donation for liver transplantation and to obtain livers which are not used for transplantation. And one category of these livers are explanted livers from metabolic disease. And I will come back to that during this seminar. I just reminded some conflict of interest. As you know, I am a pediatric hepatologist at Clinique Saint-Luc at Brussels, University of Louvain. And I have also a research lab at the University of Louvain. I am heading the liver tissue bank at Clinique Saint-Luc. And also to develop these advanced therapies, I did found a company, biotech, which is called Cellion, for which I have been initially CEO and CMO. This is the company which is organizing the clinical trials. But all what is tissue development and ITMP development is in the hospital. The history was hepatocyte transplantation. Many of you remember that in the years 2000, a number of hospitals in Europe, including Brussels, London, Barcelona, and others, have developed hepatocyte transplantation. And the idea was really that in children with inborn error of metabolism, you could infuse hepatocyte in the portal vein, bring these hepatocytes in the liver, and bring de novo enzyme activity to improve the metabolic control of these children. And this therapy has been, to a certain extent, successful, especially when we were using freshly isolated hepatocyte, and a number of different diseases, such as Crigler-Najjar, urea cycle disease, have been treated. So that was really the initial development of cell therapy, hepatocyte transplantation. However, we must admit that this therapy has not really been sufficiently efficient to meet the expectation. First, the hepatocyte, you need to have one liver to isolate hepatocyte to transplant one child. You are not transplanting pure hepatocyte, so this is an issue for pharmaceutical product because it's not a pure cell composition. In addition, you can be successful with freshly isolated hepatocyte, but hepatocyte do not resist very well to cryopreservation. So, and the durability of the effect was around 1 year, sometimes a bit longer, but not lifelong as we can expect with liver transplantation. In addition, you need to replace at least 10 to 20% of the parenchyma to really change the metabolic control of these children. And in addition to that, a new regulation in Europe did appear, and it became really a pharmaceutical product. There was a GMP environment required for isolation. So this has really limited the expansion of this technique. However, what we did discover, and I will come in a minute, is that by doing this quality control of hepatocyte, you can extract from the liver stem cells, and these stem cells are not aiming to replace hepatocyte, but they have a very powerful paracrine function, and these cells can be extracted from discarded liver tissue. So what did happen is that when you isolate human liver, you obtain these suspension liver cells, which is a kind of soup of cells. And when we put these cells into culture condition, the hepatocytes do not survive very long in culture condition. They die, but what you can observe is the emergence of these elongated cells, which are of the category of mesenchymal stem cells. And what is amazing with these cells is that you can expand these cells really importantly in vitro. So with one liver sourcing, you can produce a very large quantity of these cells. These cells are very specific because they have a combined expression of hepatocyte marker like albumin and alpha asthma, but in the normal liver, you cannot find cells which combine the expression of these two markers. Either you have positive asthma cells or positive albumin cells, but you have no cells combining these two mesenchymal and epithelial markers, while these cells that you obtain in vitro do express these dual markers. And in fact, mesenchymal stem cells, which are also now called medicinal signaling cells, to insist upon their capacity to produce a number of active cytokines, can be extracted from a number of different tissues. Most commonly, these cells are extracted from the bone marrow, and they are used to develop medicinal therapies for graft versus host disease or for bone or cartilage replacement. Cells can be extracted also from cardiac tissue, from adipose tissue, from amniotic fluid, and from the liver. And this is the particularity of the cell that we have developed, is that it is extracted with the liver with some specificity. One major specificity is that they do not express HLA class II, which is, of course, very important when you infuse cells to patients. And there are a number of clinical applications that are being developed around the world. One of the most common one is graft versus host disease, and this was the original discovery and development in Karolinska by Katharina Leblanc. When you have patients with severe graft versus host disease, you infuse to these patients MSC derived from a third donor, not the donor, not the recipient, but a third party. And you can, as they call that, reset the immune system, because these cells have a very strong immunomodulatory potency. And this has been developed also in bone disease, Crohn fistula. Takeda is commercializing now bone marrow-derived stem cell, mesenchymal stem cells, that is applied in Crohn fistula, and which has received, of course, market authorization in this indication. So this is becoming clinical reality. The cell that was extracted from the liver is quite unique, because initially, like hepatocyte, we did infuse these cells in the portal vein, but you understand that this is quite tricky. You have to access the portal vein, and this is difficult to repeat that several times if needed. But what was shown is that when you infuse the cells by peripheral vein, these cells do not persist in the lung. They just cross the lung. There is no lung entrapment, and these cells home to the liver. There is a major homing to the liver. More than 60% of the infused cells reach the liver. Some of them reach the bone marrow, the spleen. But what is also very interesting is that when you infuse these cells, there is a homing in inflammatory sites. And this was the case of this patient with hemophiliac arthritis. There was a spot of cells in the left ankle of this patient. So this is really what is exciting, is that these cells, which do have a very strong anti-inflammatory immunomodulatory activity, are homing to the liver and homing to inflammation. And that's why we started to dream that these cells can treat patients with severe inflammatory liver disease. So the cell is called hepastem. It's a stem cell coming from the liver. And I have to mention that when you see this logo of Celion, it means that the work has been done by this biotech company. And what they do, of course, they obtain the suspension of liver cells. These cells are plated and expanded in vitro to produce these MSCs. And so far, this was started first in children with inborn error of metabolism, because at the beginning of this development, we were considering, like many others, that MSCs could differentiate into hepatocyte, cardiomyocyte, chondrocytes, bone tissue, but this is not the case. But anyway, there was a clinical trial in children. There was some benefit. It has been published, but not again sufficient to replace liver transplantation. But what is important is that now we have children who did receive these cells more than 10 years ago, and that there is a very strong safety package. None of these children have experienced adverse event following the infusion. As I mentioned, initially we developed this therapy to treat inborn error of metabolism. But after that, following the discovery of paracrine activities and immunomodulatory properties of the cells and anti-inflammatory properties of the cells, we did switch the indication to treat patients with inflammatory liver disease, mainly NASH and ACLF. And currently, ACLF, acute and chronic liver failure, is the first target indication. So it has been shown, and there are many publications showing the potent anti-inflammatory activity of these cells. When the cells are facing pro-inflammatory condition, which is the case in acute and chronic liver failure and in NASH, there is a boost of the anti-inflammatory activity, which is here IDO activity, secretion of prostaglandin E2, but also hepatocyte growth factor is produced by these cells, and this, of course, plays a role in regenerative disease. Because in liver inflammation, it doesn't mean that the hepatocytes are lost. The hepatocytes are just blocked. The function is blocked by the severity of inflammation. And the idea is really to control inflammation, to allow the recovery of functional activity, which is kind of paralyzed by the cytokine storm and the strong inflammatory condition in these diseases. The difference between the cells derived from the liver is normally one of the definitions of mesenchymal stem cell is that they can differentiate into adipocyte, osteocyte, and chondrocyte. This is not really the case of the cells coming from the liver, which is already specific to the liver and is no more easy to differentiate into this tissue. They do express the classical mesenchymal marker. They have a strong anti-inflammatory immunomodulatory properties, but that is the same as most MSC. But importantly, they do not express class 2 antigen, and they have very strong hepatocyte growth factor secretion. So these cells, when you infuse these cells in patients with inflammatory liver disease, they reach the liver and they produce this potent secretome, which includes these cytokines I already mentioned, TG2, IDO, TGF-beta, also hepatocyte growth factor. Another strong activity is S1P, which is reinforcing endothelial barrier, and we know that in cirrhotic patients there is endothelial leakage. And in addition, although these patients are probably too far in the evolution of cirrhosis but there is some demonstrated antifibrotic activities in animal models with F2 fibrosis. And again, as mentioned, the cells can be easily infused by peripheral veins, so it's perfectly tolerated. There is no more difficulties to reach the liver by the portal vein, but a simple peripheral infusion. So currently the first indication is acute on chronic liver failure, and there is an ongoing trial in Europe which has already recruited 92 patients. The total number of patients with ACLF for this study is 130. And this is ongoing in 17 European countries, 52 participating centers. There was also a phase 2A study in NASH. The idea in NASH is really to bring back these patients from steatohepatitis to simple fatty liver disease, to stop inflammation. And there are animal models showing that. And of course, later on, there are other possibilities to treat less severe patients like acute decompensation. Urea cycle disorder, this was, as I mentioned, the very original development. These patients are just now followed for long-term safety to reinforce the safety package of this therapy, which is, of course, the first concern of everyone and of the regulatory authorities. Now, what is, you are probably familiar with acute and chronic liver failure. These are currently adult patients, and you know that it has been also described in children, but we start here in adults. They have sometimes even unnoticed chronic liver disease and fibrosis and cirrhosis. Then suddenly they become jaundice, they have this acute decompensation, and ACLF is the next phase when you have on the top of acute decompensation secondary organ failure, like the heart, like the lung, the brain, the kidney. And this is what is called acute and chronic liver failure. It's a very severe disease with more than 50% mortality after 90 days. And there is no treatment beside a standard of care. And most of these patients do not have priority access to liver transplantation. And that's why there is still a very important mortality. And this disease is really related with a cytokine storm. There is a strong inflammation with inflammatory marker in these patients. So the first trial, the phase 2A, was of course dose finding and safety. And this has been, of course, achieved. There was no serious adverse event. And there were interesting signals in the patients is that there was a trend to an improvement of MELD, Child-Pugh score, bilirubin level, but also inflammatory markers, which are also a landmark of this disease in adults, CRP and leukocyte count. So, of course, this is not a controlled trial, but we did match the patients from this first study with patients from a European database, which is called the CLIF database in Europe, organized by EASL. And we at least showed that the survival in these patients was comfortable as compared with this database, but we have to be very, very careful. Of course, this is not a controlled trial. This is just extracting matched patients from real-life data. It cannot be considered as a control. But anyway, this led the company to go for the next phase clinical trial, which is the one ongoing currently, is that the patients who are admitted for acute and chronic liver failure, first they have a 7 days period, and this was a request from the regulatory, from EMA, just to avoid to treat patients who could recover spontaneously. So if they persist in ACLF for 7 days, then they receive 2 infusions of the liver-derived stem cells 7 days apart. And the primary endpoint is very strong. It's survival. Either a patient survives or they die, and the idea is to improve the survival. Once you survive beyond day 90, usually then this acute phase is over and you go back in a kind of chronic cirrhotic compensated disease, and the mortality then is around 3 to 5% per year, like cirrhotic patients. But then, of course, you can decide and treat patients. It can be considered as a saving or as a bridge to transplantation. So these are the different countries where the trial is ongoing. As you see, many European countries, and we are now developing the product also in Argentina. One of the reasons is, of course, to improve the recruitment, which is always very challenging in clinical trials. So now, how is it manufactured, this but we need to have first tissue, liver tissue. Not much, because with one source liver, we can treat many patients. And these are either reduced size liver, sometimes unused splits, but what we have developed is the use of explanted metabolic livers. From the ones which are histologically normal, there is one single enzyme defect, but the paracrine properties of the cells are completely preserved. And these are excellent quality livers because they come from young patients who have no toxic injury, no viral disease. So this is an excellent, and in addition to that, it's a rewarding experience for the families because these livers are not just destroyed. So this was in my hospital in Clinique Saint-Luc. My colleague, Professor Rodin, de Manier, Tambucci, did explant the liver from a patient with metabolic disease, and this liver is then shipped and treated up to a liver cell suspension, and this is cryopreserved. And these cell cryopreserved are then thawed, expanded in culture condition, placed in vials, and these vials are again cryopreserved, and they are shipped to the patient, and there is a simple reconstitution, mixing the cell with a diluent, and it can be then applied. So with one single liver, hundreds of patients can be treated. So that's, of course, the benefit of in vitro expansion of these cells. So this is interesting. Professor Rodin, when he did prepare this liver, was showing me this Cantley line. This was a surgeon from the beginning of the 20th century. And when you perfuse here the right portal vein, you see this line, which is in fact separating the left and right lobe. And in fact, what we do is just to prepare the liver as if it was due to be transplanted, and then this liver will be perfused with collagenase. So this is again what is of course important is that there is no waste of liver tissues, no competition with liver transplantation, and that these livers can be used to treat patients with lethal disease. Of course, this is still clinical development. It's not yet an approved product, but it's a product in advanced clinical trial. So many people are asking, well, what about the legislation? And we are not very familiar with legislation on tissue. We are familiar with legislation of organ. But there is a very well established regulation in Europe, and the obtention of liver tissue or tissue in general is under the human body material regulation and advanced therapy medicinal product regulation, and each country have, of course, applied this in the national law, and this is the case, of course, in Belgium, when there is a specific legislation on human body material. So the tissue must be transferred to a tissue bank. A tissue bank is not a biobank. A tissue bank is dedicated to use tissue for clinical application, not for research. Biobank is collecting tissue for research. Tissue bank is collecting tissue for clinical application. And of course, you need to have ethical approval, and you have to explain the ethical committee what you will do with this, and including the transfer to, here in this case, a biotech company, which is processing the material, because you need to have of course, also manufacturing authorization under GMP to manufacture advanced therapy medicinal product. And of course, the tissue bank is in charge of guaranteeing the safety, anonymity, control of the use of the tissues according to EU and Belgian laws, and of course to have the approval and informed consent of the donor. So just to summarize, in fact, and the idea of this webinar was not only to show the development with advanced therapy medicinal product, but also to highlight the fact that sometimes we just think that some liver tissue are useless, but this liver tissue can be used to prepare this advanced therapy medicinal product. And this is just a summary of the specificity of this product, because it is liver derived. This is a bit unique. Most commonly, it's derived from the bone marrow, from adipose tissue, which easy access. But with one liver, you have a huge quantity of these cells, while with one aspiration of bone marrow, you have much less quantity. And now the simplicity of intravenous infusion. And now the safety. We have infused children in the beginning. Now we are infusing adults, but more than 100 patients have received already. There is what is called in this clinical trial the DSMB, Data and Safety Monitoring Board, which are, of course, looking and watching the safety of the clinical trials. And for this trial, as I mentioned, 92 patients have been infused so far. And after 25, 50, and 75 patients, the DSMB gave the green light. So the idea is really of this ongoing study to confirm the preliminary finding found in phase 2A and to have the proof of concept that MSCs derived from the liver can indeed control inflammation and allow the recovery of acute and chronic liver failure.